Alright, so this is going to be the first video of the series about option E, which is on paper 3 of your IB exam. But notice that I'm not starting with E1, I'm starting at E2, which is the perception of stimuli. So we're going to kick off with E.2.1, which really is the overview of what a stimulus is and how we perceive that with receptor cells. But then we're going to focus in on the eye and vision, and then we're going to focus in on the ear and sound and how we perceive sound. So just as a quick reminder, that a couple of things that you should review before really moving forward are our nerves or neurons, the central nervous system and the peripheral nervous system, action potentials, and resting potential. Resting potential. Additionally, you should know how a signal is propagated down an axon, and then also synaptic transmission. So what I'll be talking about in this video is based on your understanding that you already know this, that you understand what's going on and how signals are sent through your nervous system around your body. Okay, so that is the given assumption. So let's go ahead and move to E.2.1. And again, this is outline the diversity of stimuli that can be detected by human sensory receptors, including mechanoreceptors, chemoreceptors, thermoreceptors, and photoreceptors. And so just want to make a quick point to identify the command term of outline. And so really what we need to do is that. It's not just a list, it's an explanation, it's providing an example or a location and a function, and those are all things that are required for our answer here. So we'll go ahead and, why don't we go ahead and make a chart that explains some of this. So let's do one column is type of receptor. We've got another that is function, and we've got example. Actually, I'm going to scoot that over just a little bit. Why don't we scoot this over? Function, and then example or location. Okay, so the first one is the mechanoreceptors. And the function of these mechanoreceptors is to generate nerve impulses when stimulated, which is really the function of all of them. But we're going to go ahead and write that here. Generate nerve impulses or action potentials when stimulated. And that stimulation may come in the form of touch, pressure, stretch, or balance. So, for example, there are changes in for changes in pressure. There are receptors in the skin Well, I guess we can put that. Oh, sorry. We can have changes in pressure, texture, or vibration. Let's go ahead and add that. We'll put the skin over as an example. So, an example would be for pressure, we have pain, touch, and tension receptors in our skin. There are touch receptors in, there's also for vibration, we have the inner ear, which we'll learn about in a little bit later, and balance or equilibrium. I guess sometimes it's referred to as equilibrium, but really your ability to stand up straight is involved here. So that's one type of receptor. Oh, I'm running out of space here is what I see. So let's see, pick a different color. We're going to talk about chemoreceptors. Chemoreceptors, and their function is to respond to chemicals in the environment. So specifically solutes and vapors. To solutes and vapors. For example, some of these solutes could be taste on your tongue, or even the amount of CO2 in your blood, which is really kind of like a pH sensor, because CO2 makes solution more acidic. There are chemoreceptors to measure the amount of carbon dioxide to trigger your body to expel some more of that carbon dioxide through your lungs and through exhalation. And then there are chemical vapors, which are, essentially, think of smell in your nose. Okay? So these are all ways that we sense the environment around us, and all these signals get sent to our brains. So I'm going to go ahead and erase these two because I've run out of space, but make sure you jot these down, and then you might need to hit pause, jot them down, and then hit play again. So third, we've got thermoreceptors that respond to changes in temperature. An example would be these thermoreceptor nerve endings that are found in the skin to regulate our temperature. So think back to temperature regulation that we talked about in the body systems unit. I would actually urge you to go back and look at your notes on this body systems unit to ensure that you can see the connection between these. And then lastly, the fourth group are the photoreceptors. And these actually are used to detect electromagnetic radiation. And the examples are the rods and cones in the retina of your eye. Okay? And so those are the four types of receptors that we have in our bodies. You should know that there is some regulation and there's some range for these thermoregulators. For example, there are wavelengths of light that we can't see, other organisms can, but they do help us make sense of the world and help keep the brain aware of what's going on in the environment, in the outside world, sometimes even the internal world, okay? So that should cover E.2.1. All right, so the other thing that you need to be able to do is label the diagram of the structure of the human eye. Again, label. You don't have to draw. You need to be able to label it. So, um, I guess if we should go ahead and get started and do this, huh? All right, so let's start with a couple of let's start with E here as the pupil. Or maybe I can pick a better color than this. That's not what I want to do. I apologize. E is the pupil, and this is a hole or entry of light to the eye. I'm not going to take the time to write down the function. I'm running out of space, but you have that and it's in your book. You are responsible for knowing this. So we've got the pupil. We've got, let's see, here we've got our lens, which adjusts the focus of the light. Here we've got our conjunctiva. So this is basically a protective layer around the eye to protect it from damage. We've got the iris right here, which is pigmented. This is where your eye color comes from. It regulates the entry of light. This down here is your eyelid, which is kind of a strange drawing of it. We've got the retina, which we'll focus in on just a moment. Back here, which is where the rods and cones are, the photoreceptors. We've got the choroid, the fovea, and this doesn't have a label to it, but this is the optic nerve. This is the blind spot, because there is no retina right here, because that's where the optic nerve hits the eye. You have the sclera here. We've got the ligament, which is the muscular part of the iris. We've got the cornea. And we've got the aqueous humor, which is just the fluid within the eye that helps light pass through. So here are the things that you need to be able to label. That's all this is asking you to do. But I would tell you that you also need to be able to annotate with functions. You need to be able to tell me what's going on here, because the next target has to do explicitly with annotating a diagram of the retina to show the cell types and the direction in which the light moves. So we're going to move to that, and then compare rod and cones, and then talk a little bit about some optical illusions. And as we move to the retina, I should just point out that when we're looking at the diagram of the retina, we're actually focusing in right in this section right here, so that we're seeing a little bit of the ganglion cells here, and then the retinal cells and the cones and rods in the back. Okay, so let's move to that diagram. I've already inserted it in here. So you'll be able to annotate a diagram of the retina to show the cell types and the direction in which the light moves. So some of these have already been labeled in this diagram, but you need to be able to annotate them. So here are the ganglion cells. Over here on the right, we see that light is moving into the eye this way. So the iris and the cornea are all over on the right-hand side. Light's moving in through the ganglia, through the bipolar neurons, and then to the photoreceptors, to the rods and the cones. But that signal is actually hitting these cells in the back, the retina, the choroid in the back, and it's then sending a signal to the rods and the cones, which then propagate and create a synapse with these bipolar neurons. So these rods and cones, these are cones up here and these are rods. These are the photoreceptors that we just talked about in E.2.1. These photoreceptors create a synapse with the bipolar neurons. These bipolar neurons, notice that with a cone, can actually make several different connections, different synapses, but each cone is only going to one bipolar neuron, as opposed to the rod cells in which several rod cells can actually join with a synapse. And that bipolar neuron creates a synapse with the ganglia, which are the body or the soma of the optic nerve, and the ganglion is going to take this electrical signal all the way to the brain, so the brain can process what's going on. So you need to be able to add what I said to this diagram, okay? And it's pretty straightforward. But again, light's coming in from the right to the left, and it's hitting the back of the eye, and then it's creating electrical and then chemical, then electrical and chemical and electrical signals so that the brain can then process what it has seen. Okay? So we need to be able to compare rods and cones. Compare rod and cone cells. Okay? So, again, this isn't terribly difficult, but it's another list of things to memorize. So, again, it says compare. So, you should know that this is asking you for similarities and differences between rods and cone cells. So, let's make sure we answer this question in an easy enough way that will help us make sense of that, and to convey that we understand the difference. Rod cells and cone cells. Okay, so I got my two columns here. So these rod cells work best in dim light, as opposed to so we can say that they are sensitive to dim light. They actually don't work well in bright light, whereas our cone cells, as a difference, are sensitive to bright light. Okay? As far as the types of rod cells, there is only one type of rod cell, but there are 3 types of cone cells. That's right, 3 types of cone cells. So we're going to talk about those in just a moment. And another point here is that the rod cells are concentrated around the fovea, whereas oh, scratch that, I apologize. I'm getting my cells mixed up. I'm glad I caught that. The rod cells are throughout. The retina, and the cone cells are concentrated around the fovea of the eye. And the rods are responsive to all wavelengths of light, well, let's say all visible wavelengths of light. Um, whereas remember there are three types of cone cells. So the short ones, the short rods, um, can see violet light. The medium ones can see green, and the long ones can see red. For those of you who have taken physics, think about the wavelengths of these colors in nanometers. And I hope that you make a connection there. Another thing here is that many rods can connect with one bipolar neuron, whereas cones, let's say one cone connects to one bipolar neuron. And that's a pretty good long list comparing the two types of cells. All right, so E.2.5, which is the last bit about eyes, is basically how we understand the anatomy of the eye and its functioning in what we can often experience in optical illusion. So the first thing that so it says to explain, so you need to provide some explanation, some rationale, some cause for some of these things. So we need to talk about first about edge enhancement, and then we'll talk about contralateral processing. So in edge enhancement, there's the textbook actually has a very good diagram called the Herman grid. The Herman, I don't know why I put a space in that. The Herman grid illusion. So if you look at that, it's in the textbook in this section of the reading. The idea is that if you focus on any one part of the illusion, you'll start to see gray spaces at the intersections of the white and the black. And the idea here is that because of what we understand about where your rod and cone cells, photoreceptors are, and because cones are sensitive to bright light and happen to be centered or concentrated around the fovea, or the center of your retina, wherever you focus, those are where the cones are receiving most of that light. So if you're focusing at one corner of this illusion, your cones are not receiving the signals they need to do this. So really, what you're seeing at the gray sections are part of your peripheral vision, which cones are not a part of. So they're not able to detect that fine acuity that the cones are able to help. And so therefore you're relying on your rod cells, which don't have as high of acuity or accuracy. So, and in fact, many scientists think that those neighboring cells get turned off. And so instead of actually perceiving these spaces as white as they actually appear gray. But essentially it comes down to that your rod cells are not as fine, they're not as good at detecting contrast, or because they're not triggered by bright colors, but rather dim things. So those rod rod cells are not going to be able to make that distinction between white and black. In fact, what they're going to do is kind of make them a little fuzzy. Okay, so that's the edge enhancement. So really, when you focus on something, those cone cells are going to help us determine that clear edge between light and dark, find edges in objects, and perceive those stimuli better. So with contralateral processing, essentially the idea is that we use both of our eyes to receive signals from our environment, but those signals actually become inverted images on the back of our retina that then, remember, get sent, that image or that impulse gets sent to the brain, where the brain has to make sense of it. So these inverted images from the left side and the right side have to be sent to the brain, and in order to make meaning of these images, both sides of the brain have to be used, must be used, and information from both eyes has to be processed. So contra meaning against or opposite, and lateral, talking about the direction in which the brains are functioning. So we've got contralateral processing in order to make meaning. And there's a good example of this in your book at the end of this section about eyes. All right, so very quickly we're going to do E.2.6 and E.2.7 because I realize that this podcast is quite lengthy. So this first part right here, excuse me, sorry about that, is the pinna, or the outer ear that funnels sound into the ear. That sound then finds its way to the eardrum, or tympanic membrane. Sometimes it's called the tympanic membrane. That eardrum then shakes and actually makes these middle ear bones move. They make them move, rattling this oval window. If let me just make sure. Yeah, the oval window. Just had to make sure it wasn't the round window. The oval window looks more oval than that one, I guess. The oval window, which then transmits vibrations from these middle ear bones into the fluid of the cochlea. So this cochlea, which is here, is filled with fluid, and the oval window adds some pressure waves into that fluid, and then it moves the hairs within that cochlea, which then release neurotransmitters that will then be sent to the cochlear nerve, and ultimately to the brain. Okay? Yeah, so let's label a couple other things. These are the semi-lunar canals, which are related to balance. That helps us with our standing up straight or knowing when we're falling, I guess. Maybe a good way to say that. And let's see. Let me make sure I've got everything that we need here. We've got the eustachian tube. Oh, the cochlear nerve is sometimes also referred to, IB sometimes refers to this as the auditory nerve. Maybe that's an easier way for you to remember that. It's the auditory nerve that carries audio through the ear. So that's what you need to be able to label this and also explain how sound is perceived by the ear, including role of the eardrum, bones of the middle ear oval and round windows, and the hair cells. Now, the round window dissipates the vibrations so that the pressures of the fluid in the cochlea don't continue to send that signal and overload our brain, but instead they dampen that signal once it has been felt. Okay, so be able to explain how the ear functions with that, and as you can imagine, it'd be very problematic if the eardrum ruptured, because then it wouldn't be able to send that signal to the middle ear bones, to the cochlea, to the hairs inside the cochlea, to the auditory nerve. Okay, so that's what you got to be able to do, and that's all for E.2.